the Faculty of Science and Technology. I'm also a member of the Science Outreach Committee. So I'm really pleased to be able to be here tonight to introduce our guest speaker tonight, and that is Dr. Shawn Lewenza. Dr. Lewenza is an associate professor in the Faculty of Science and Technology at Athabasca University. He completed his PhD at the University of Calgary in the Department of Microbiology and Infectious Diseases, and is still an adjunct member of that department. He also completed postdoctoral fellowships at the University of British Columbia, as well as the Pasteur Institute. I've actually known Shawn for quite a number of years. So I've known many of his research endeavors over the years, and it's been really exciting to have him with us at Athabasca University. Shawn's research is mostly focused on understanding how environmental bacteria like Pseudomonas aeruginosa cause chronic infections. And so this is through focusing on understanding how Pseudomonas aeruginosa is resistant to antibiotics and how they form biofilms that are very antibiotic resistant and help evade the immune system. He says that his guiding hypothesis is to try to identify how and what bacterial genes are turned in a biofilm and turned on in a biofilm and how they contribute to long-term survival. And this approach has led his team to discover new antimicrobials that block biofilms, therefore enhancing the activity of current antibiotics as well as the immune system. Shawn's recent work has used this same overall approach to study bacteria isolated from oil sands tailings ponds and to learn how bacteria respond to the pollutants present in the wastewater. This information allows him and his research team to identify bioremediation genes, which are necessary for bacteria to degrade these pollutants. It also helps them build new biosensor technologies that provide a simple, rapid, and inexpensive monitoring system to detect pollutants in the water. So I know it's going to be a very exciting talk tonight. Shawn's already achieved a number of exciting outcomes from his research, and I'm really happy he's able to share this with you tonight because the significance of his work is huge. It has a lot of potential for us in how we monitor the environment and deal with environmental change as well as climate change. So I welcome Shawn, and I'll turn it over to him. Thank you.
Thank you, Shauna. So the only warnings I have are I'm not an oil sands mining expert and I'm not an oil sands microbiology expert, but that's the kind of things we're going to talk about today. And the title is The Good, the Bad, and the Ugly of the Microbes in the Oil Sands Tailings Ponds. So I will talk a little bit about the mining process and about how the tailings ponds are formed and what's in them. And then I will also talk about this microbiome concept, also the metagenomics, which is this new science of studying bacterial communities. So we'll go over some of the darker sides of the oil sands mining process, but also there's a lot of good and there's a lot of potential that exists by harnessing the microbes that are within the tailings ponds. And so that will lead me to talking a little bit about this new technology that we produced, these bacterial biosensors. They are a new test for naphthenic acids in water that is quite simple. And actually, it's something we just patented. And I think it's the first patent at Athabasca University. And then I will talk about the use of these biosensors for identifying bioremediation genes. So when we think about the oil sands mining, at least when I think about it, usually it's something that's happening on a grand scale. It's these large pieces of equipment moving the oil sands through the mining operations. It generates these large mining sites and tailings ponds, things that you can see from Google Earth Maps. And instead here, I want to focus more on the micro, which is zooming down into the water of the tailings ponds where all the microbes live, the microscopic organisms that have a huge impact on this environmental problem of the tailings ponds. And so we're kind of changing our focus from the macro to the micro for this talk. So just a brief review of what they do when they mine bitumen or oil from the oil sands. So starting in the top left, beneath the layers of the topsoil are the oil sands, which are physically mined and transported by these large trucks, and then are broken up into smaller pieces into a finer sand. And then basically the sands are mixed with a hot water. It's also alkaline, so it has a basic pH. And this is the simple way of extracting, first creating a slurry, and the initial stages of extracting the oil out of the sand. In the next stage, you have these separation vessels where the different components of that mixture separate: the sand at the bottom, the water in the middle, and then the oil and froth at the top. And so the sand can be separated out from the bottom. The water can be removed, and as can the oil that's recovered through the froth in the bitumen. Now there's several to skim off that top layer, and there's several other stages of sort of processing it. But essentially it's using water to extract the oil from the sand. Now there's three major problems with these are some of the major problems with the oil sands mining. First is that it uses a lot of water. You need 3 barrels of water to extract a barrel of oil from the sand. It produces these tailings ponds, which have numerous contaminants that are toxic, that are very large. And, they need to be, ultimately the water needs to be treated and the land needs to be reclaimed. And it is a greenhouse gas intensive process, the drilling or the mining and extraction of bitumen from the oil sands. Now, given there are these problems, I am aware now more and more of all the research that the oil sands industry is doing to mitigate or reduce all of these impacts. Significant uses of recycling of water to limit the amount of fresh water that we use. Lots of work in the area of remediation and figuring out how to treat the water, treat the soil, at the end stages of the mining, and always an effort to reduce greenhouse gases, and even more so in an economic climate where gas prices or oil prices are low, like now. It drives innovation, and it makes more effort to mitigate and reduce costs and do things more to eliminate water use and decrease greenhouse gases, etc. So if we look a little more closely at the oil sands ore and the tailings, the sands themselves, the cartoon at the top right here, you can see the bitumen is sort of surrounding these grains of sand. There's larger sands and small clays. There's small bits of water, but essentially in the sand, it's 10% bitumen, and it's mostly sand and clays and minerals. And there's a similar diagram here showing the separation of the tailings from the froth. And again, the froth contains the bitumen, which is extracted. But everything else that's in the sand goes into the tailings. So the water will extract everything else that's naturally present in the soil. And then that goes into the tailings. And those separate into multiple layers over time. Where at the top, you have oil sands process affected water. And then you have increasing amounts of tailings as you go deeper into the tailings ponds, and the base would be sand. So microbiologists love getting their hands on these kinds of samples to do all kinds of experiments, but mostly to study the microbes that are present and growing and thriving in this environment. Okay. Also just shown on this diagram is that it's not used once, the water, it's actually recycled. So the water from the ponds can be taken off again and reused. So again, hot water or an alkaline extraction reused to re-extract. And in that way, it actually concentrates all the other compounds that the water extracts through this process. So that's why lots of contaminants and organic compounds and metals do accumulate in the OSPW. So we can start off with some of the more harmful side effects or impacts of the oil sands mining, which, there is a loss of boreal forest. It's a greatly disturbed habitat for wildlife. There's the use of fresh Athabasca river water, the generation of large volumes of wastewater that's stored in tailings ponds. And ultimately, it needs creates a requirement for us to treat that water and reclaim the land. But also there's potential for the leakage of the toxicity from the tailings ponds water into the ground. And also there was harmful emissions coming off tailings ponds, hydrogen sulfide gas, the volatile hydrocarbons, and methane. How much land is being used? This is one of the topics where I see different areas, different numbers from different perspectives. These are the numbers from the Canadian Association of Petroleum Producers. And you get different information if you're talking to reading the literature from the Pembina Institute or from the oil sands producers. But as an example, there is 142,000 square kilometers of oil sands in northern Alberta. Most of it, again, is many meters below the surface. And then the mineable area is 4,800 kilometers, but the actual mined area, so the actual space being used, is less than 1,000 square kilometers, or 0.2% of the boreal forest. Now, it does produce very large volumes of wastewater. And as I mentioned, you can see this mining sites and tailings ponds from Google Maps. So the volume is estimated at about 1 billion meters of wastewater. And just, we reuse much of it, but if the oil sands mining proceeds, it's going to generate more water. And it's just currently stored in tailings ponds estimated at around 175 square kilometers. But this entire area of mining and tailings ponds is around 900 square kilometers. So there's no release policy, which means that wastewater is not released it's stored in tailings ponds, and it's stored generally till the later stages in mining, or until the methods of treating the water have been worked out. And these are things that are being worked out at this time. There are tailings ponds that are remaining inactive and are sort of experimental systems for how to treat and remove some of the key compounds that accumulate in the tailings ponds. That, again, they are originally naturally present in the oil sands themselves. Here's a sort of government drawing of some of the engineering of building these tailings ponds with an effort to capture anything that drains and leaches or it's leaving tailings ponds. Now, sometimes there's an argument that there's a very solid clay-like bottom due to all the settling of all the fine tailings. But if water is settling into the ground, there's different catchments where they collect seepage, they pump it back into the tailings ponds. There's various monitoring systems and additional sort of engineering walls and things to prevent this water from getting back into the Athabasca River or into the ground. But those are the risks, that this water might leak into the ground or river water. There are occasional breaches, tailings ponds. And, this is a large area where it's not being released. And so for the time being, this is just an environmental hazard. What are the contaminants of concern? So there's a long list of things that are in the water. At the bottom here are all the metals. And in order for any of these compounds to be considered needing treatment, they need to be above a certain threshold. And this is data from the Canadian Oil Sands International Alliance. So you'll see in the gray, there's lots of treatment not required. So you may have metals here that are at milligrams per liter is parts per million. So the very low end of this range is very low levels of parts per million, or in this case, cadmium parts per billion. But what I'm interested in and what's the most abundant compound is this. Here it's referred to as acid extractable organics. These are the naphthenic acids. They're the most abundant, and they are considered to be among the most toxic of the compounds in the tailings ponds. That's where most of the effort is being spent, on figuring out how to treat the naphthenic acids. So in terms of the toxicity, it's still, it's still an open question exactly figuring out what are the toxic components, specifically in the naphthenic acids, because although they're the best studied, and they are known to be toxic to bacteria, amphibians, fish, birds, mammals, bacterial degradation of the OSPW often reduces the toxicity. So with the microbes degrade are the important components, the toxic components. But what I would like to share here is that this is a very complex family of chemicals. And that's what's shown in this mass spectrum of extracted or concentrated naphthenic acids from water. And so each compound is a line in this curve, and it has a specific mass. And the higher the line, the more abundant the compound. And the point here, some are abundant at high amounts and some are less abundant, but there are very many peaks on this graph. There are hundreds of chemicals here. And although we might know the mass and that they fit into the category of naphthenic acids, we don't actually know exactly their structure, and we don't know which ones are exactly harmful. So from a chemistry perspective, it's a very complicated and challenging problem. So if we switch gears a little bit to a famous microbiology concept, it's called the Great Plate Count Anomaly. And the Great Plate Count Anomaly means that although there are very high numbers of bacteria in a gram of soil, so they're estimated to be up to, so 10 to the 10 is 10 billion. So there's more bacteria in a gram of soil than humans on the earth. And if we try to grow them, so if we try to do this typical experiment of taking a sample that has water or soil resuspended in water, which has lots of bacteria, and we take a small aliquot and we plate it, we expect to get high numbers of bacterial colonies. However, we only recover, well, the initial estimates were 1% of bacteria that's in soil. So we can't grow most of them. And the why is that either we don't have the right growth conditions, or they need to grow in relationship or community with others. And although it's still true that we cannot mainly grow all the organisms, let's say in any environment, let's say whether it's a water sample or whether it's a fecal sample studying the human fecal microbiota, there are reports where we are getting better at it. And in some cases, they claim to be able to grow and isolate 50 to 80% of the microbes. So for a microbiologist, that's a problem because you want to be able to recover these isolates and grow them and study them and use them in the lab. But if they exist in the water soil and you can't grow them, that's a challenge. So the next important thing that's been happening over the last 10 years, and it's the study of the microbiome. And the microbiome is simply the community of microbes in any environment. And here in this picture is just a representation of all the microbes that live within and on and in the human body. And so studying those organisms and knowing who's there, which microbes are there, the diversity of microbes has basically created a huge revolution in microbiology. It's true for human health. It's true for environmental microbiology. So I'll try and explain the methods that we now know. While we still can't grow them, we know in great detail which microbes are present in any kind of sample. And so that's this concept of metagenomics. And that's basically sequencing the DNA of a microbial community. So if you have a sample with various microbes, and here you're just seeing different shapes of microbes, the colored circle inside each cell is its DNA. It usually has a circular chromosome, one chromosome. An average microbe has 4,000 or 5,000 genes on its single chromosome, whereas a human has the estimates around 25,000 genes. But if you have a sample with very low water, and you isolate the DNA from that sample, you have very little DNA. But it is enough to sequence the genomes using these modern DNA sequencing technologies. And so that you can, once you identify and put together the sequence based on the DNA present in that sample, you can identify which organism it is. That's a very computational challenging problem because you have low amounts of DNA. However, what is more successful or is more commonly used for this understanding the community is they amplify a single gene using PCR. So here is a representation of a single gene from this species A and species B and C and D and E, etc. And that gene acts as a barcode. It acts as an identity of the species that are represented in the sample. So by sequencing the single gene, it can tell you, and comparing those sequences to the databases, it can tell you the exact microbes that are present in a sample, even though you can't grow them. So I told you that the tailings ponds are thriving environments for microbes. And where do they come from? So they originally come from the oil sands themselves, right? And microbes are important in the decomposition of organic matter, in the formation of petroleum, which occurs over very long periods of time. And microbes are the main decomposers. And all the microbes in the oil sands make their way into the tailings ponds. They also add water from the Athabasca River into the extraction. And the microbes in the Athabasca River are also making their way into the tailings ponds. They might be quite similar because the Athabasca River water is exposed to the bitumen deposits in the soil on the shoreline. A very small amount of airborne bacteria. So just the point of the natural microbes in the oil sands and the fresh water from the Athabasca River, that's what makes it to the tailings ponds. And there's actually quite a lot of work recently in this area, and I don't have time to go into it, but there's a lot of work at looking at what are those communities using that microbiome approach that I told you about, sequencing that one barcode gene in the entire community. You do not have to grow them. You just need a water sample or a soil sample, and you can extract all the DNA from that sample, which is mainly from the microbes. It could be from other fungi or other microbes, but from a bacterial point of view, we can identify the bacterial community. So that water, again, it has alkaline water, clays, unrecovered oil, so a small percentage, the naphthenic acids, metals. They add other compounds to try to dilute and make the oil more viscous to process it. But what they find is in all these different layers of a tailings pond, the fresh, it's called the aerobic water, or the top layer, where all the aerobic bacteria are found. But largely it's dominated by anaerobic bacteria in the deeper layers. So really within the first meter or two is aerobic, and beneath that it's anaerobic. And so those communities have been done and assessed in various ponds that are at different stages, that are used by different companies. Some are active, some are non-active, some are at different depths, and they find different communities in each of those different variables. And in part, that's due to the gradients of oxygen that I mentioned. It's the oxygen only reaches the top layers, but also of nutrients, and all the diverse tailings create all kinds of different conditions for different microbes. So there are basically different communities in different layers. Regarding naphthenic acids, there are many degraders present there. There are many microbes that are capable of degrading the major pollutants that accumulate in the tailings ponds, both aerobic and anaerobic microbes. So here's the ugly. In addition, although the microbes there are capable of degrading some of the pollutants, they're also capable of producing methane. So methanogenesis is basically the production of methane by anaerobic microbes. It's been observed in tailings ponds. Methane is a potent greenhouse gas. There's already a large greenhouse gas production in the mining process. And here you have methane being produced by microbes in certain tailings at ponds at certain stages. And so they're degrading the diluents, the things that are added in small amounts to help process the oil and the natural organic compounds like naphthenic acids into methane. And the estimates are, it can be as high as 2 million liters of methane per day. Now, this only happens at certain stages, depending on which microbes are present and how they've treated these tailings ponds. And then the good news is they have found ways to limit methane production by adding sulfate. Sulfate encourages sulfate reducers, and they outcompete the methane producers. So there is a lot of microbiology involved in controlling the kinds of microbes that predominate in a tailings pond. So that can be manipulated. And so I'm going to switch gears now to talk about some of the one technology that we're using to try to as an alternative for environmental monitoring. So there is government mandated water testing. It used to be called the Joint Oil Sands Monitoring Program. They monitor groundwater, Athabasca River water, but those methods are complex. I showed you an example of how well they work to generate this whole profile of all the compounds that can be found in the water. But they're complex, they're costly, they're not good for monitoring large volumes with lots of samples and thinking about managing and monitoring program long term. There is an application for some sort of system that meets these limitations. So we think biosensors, bacterial biosensors, fulfill that. They are fast and simple, inexpensive, high throughput, means they can be used for many samples. They are very specific and sensitive, and they are also quantitative. So they can also tell you relatively how much of the naphthenic acids are present in a water sample, and they can be adapted for use in the field. And so the first stage of making a biosensor is culturing microbes from oil sands water. Now, I told you that we can't culture the majority of them, but we are good at culturing certain species, including the ones that we spent a lot of time working with in our lab, Pseudomonas. So it's quite easily to take a water sample from OSPW and culture Pseudomonas. And then from that stage, we sequence the genomes, which is to sequence their entire single chromosome. Here's a picture of the circular representation of the chromosome. And in this analysis, we're looking for what they call gene islands. So each of these regions where you see these red, green, blue bars are potential gene islands, which are clusters of genes that are very mobile and that bacteria share laterally. So they don't originate from Pseudomonas, from some other species. And any microbes share genes like this very frequently. And our idea is that maybe these microbes are sharing genes that are necessary for survival in a tailings ponds. So by identifying the islands and searching at the composition of the genes in those islands, that may be one way to find bioremediation genes that have been picked up between the isolates that are specifically from the oil sands water. The other approach that we use is to study the bacterial transcriptome. And that is all the genes that are expressed in any one condition is the transcriptome. So here we sequence DNA. Here in this experiment, we sequence RNA. We should be all RNA experts now with the mRNA vaccines that are being used for COVID. But just to remind you that the DNA gets turned into a message of RNA, which is a sort of intermediary, which ultimately gets turned into proteins, and proteins do the work in a cell. So if we study and isolate the RNA from a condition, from a culture growing in the absence of naphthenic acids, and compare it to the RNA that we sequence from the presence of naphthenic acids, we can identify the genes that are induced. And this is a very conventional way of identifying bioremediation genes, because microbes often turn on the genes when they need them. They're very economical. So they have a large repertoire of genes. They don't turn them on at once. In this case, they often turn on the genes they need to degrade new carbon sources or pollutants, things like naphthenic acids. So this told us all the genes that the oil sands isolates express when they're grown in the presence of naphthenic acids. And just as an interlude, this sequencing that we're talking about is one of two parts that's really revolutionizing basically biology as well biotechnology. So I've been talking about this sequencing. It's very inexpensive. And nowadays it's been miniaturized. So one version of these sequencing machines is smaller than a cell phone, and it fits in the palm of your hand. And after isolating DNA from your sample, it can be sequenced in the manner of an hour or two. It requires some manipulation and computational analysis after the fact. But you can sequence a bacterial genome in a few hours. You can do a transcriptome in a few hours, very inexpensively. And the other piece of equipment that's very common and is also accelerating lots of biotechnology and biology is the ability to synthesize genes. So you have DNA printers, and they're becoming common benchtop equipment. Both of these kinds of equipment are in standard microbiology labs, where here you can build DNA or build genes on demand. And we use this sort of synthetic DNA process as well to build some of our biosensors. So what are our biosensors? They are basically engineered microbes that can detect that can sense the presence of naphthenic acids. So I haven't shown you any of their chemistry yet, but here is a couple examples of this family of compounds that they have various numbers of rings. So the more number of rings, the more difficult they are to degrade. They have this carboxylic acid attached to a ring, and it can be varied in length. It can be branched and be a complex branched carboxylic acid. The more branched it is, the more difficult it is to degrade. Some of them have no rings. They just have this hydrocarbon with a carboxyl group. So when you grow these biosensors in the presence of this mixture, some of these compounds will get into the cell and the cell can sense them. So we've constructed these genetic circuits which enable the cell to produce an output after it senses the naphthenic acids. And so these circuits have two main features. They have what we call a naphthenic acid inducible promoter, or it's an on-off switch. This is the DNA that turns on all the genes when the bacteria are exposed to naphthenic acids. This is upstream of a gene. It's the on-off switch. And that's represented by this purple box. And if we add that sequence in front of these 5 luminescence genes, which are taken from a luminescence bacterium that lives in the ocean, those genes produce light when they're turned on by this naphthenic acid inducible promoter. And this circuit is present on a plasmid that can be introduced to any bacterium. So basically these genes are off, and once the naphthenic acids are in the cell, there are other features of these biosensors, which I don't have time to go into. Oftentimes these, there are certain proteins that bind the naphthenic acids, and when they bind them, they fall off of the promoter and leading to these genes being turned on. So this is how the microbes specifically sense them. The point is they produce light when the naphthenic acids are present. They produce bioluminescence. We can measure the amount of light, and that is telling us that there are naphthenic acids in the sample. We're just about to prepare these manuscripts and describe them in greater detail. But given the limits of our time and the audience, I won't talk too much about what kinds of genes we're using to make these sensors. But we do have a panel of biosensors that can detect a whole range of naphthenic acid mixtures. And we're deciphering that within the mixtures, these microbes can sense a single compound or a related group of compounds and not the whole mixture. So that's what's unique about this test, is that they can see signature compounds versus the microbes don't see the whole mixture. They see specific compounds, and they produce luminescence in the presence of those compounds. So this test would be where you could send your water sample to a lab, and we could do a simple microbiology assay and tell you whether there are naphthenic acids present in the water or not. We would grow these cultures in a microplate where we could grow 96 samples at a time, or 384. We would add the naphthenic acids, incubate them in this plate reader, and this plate reader would monitor the growth and the luminescence, the light production over time. And so in this experiment here, you can see how sensitive and how strong the responses are of our biosensors. So this is the strength of the light that's being produced at different concentrations of naphthenic acids. At very low concentrations here is zoomed up. You can still see the background. The background signal is here. And as soon as you add 1 part per million and 2 parts per million, you start to see the luminescence being produced at very low concentrations, and it going up in a dose-dependent manner, which means the more chemical you add, the more signal. You can see that really dramatically at these very high concentrations. They're much higher than you'd see in a tailings ponds. These are the amounts and concentrations you'd see in a tailings ponds. And at these concentrations, our sensors have a very good detection, and they can detect them at far lower levels than is present in a tailings pond. There's also a very visual thing that you can see these luminescent bacteria produce luminescence around a disc on an agar plate where we've added naphthenic acids. So you can visually see the luminescence being produced by microbes on an agar plate. So that technology is useful for testing water it's useful for testing large samples, large numbers of volume, repeat long-term testing. We're hoping we can convince that industry to start using a biological assay instead of the analytical chemistry. But we also want to use it to try and help make progress into understanding the bioremediation, which is those microbes that are in the tailings ponds that are capable of coming to the rescue and degrading some of those pollutants. The interesting thing about a tailings ponds is it selects for microbes that are capable of bioremediation. It means that only the microbes that can survive there can eat the compounds that are present there. They are the only ones that can survive. So nature's selecting for the microbes that can do the job. And I don't have time to go into the decades of research that show very impressively that there is very numerous, large numbers of studies that show degradation of naphthenic acids by numerous isolates recovered from tailings ponds, whether they're single species, mixed species, grown in biofilms. They can readily degrade the more simple structures. It's a little more difficult to degrade the more recalcitrant compounds. But moving forward, we're now trying to answer, how do these microbes degrade Na? What are the pathways and genes in these aerobic and anaerobic microbes that can degrade naphthenic acids? And ultimately, how can we use that information to optimize this bioremediation process, which is one of the leading candidates in how they will be able to treat this large amount of water and to remove compounds like naphthenic acids. Now, for a long time, biochemists and oil sands microbiologists have been able to propose these pathways that take a naphthenic acid and it gets degraded through this sort of biochemical pathway into these more simple compounds that the microbes can integrate into their main pathways to produce energy. And each step of the way requires a bacterial enzyme. And that's the missing point, the missing information, the gap in our information is which enzymes do this. And that's the kind of thing we are trying to figure out with our biosensors. So I'm getting near the end and just double checking the time here. So we want to screen. It would be great if we could take a water sample from the tailings ponds and grow all these hundreds of species, and we could easily test them with our biosensor in a way that you couldn't really do very easily with the analytical chemistry method. But as I mentioned, we aren't really able to grow those species. So what we do and what we can do is we can capture all the DNA that's present in that sample, which is representative of all the microbes, and we can clone it into small, manageable sized fragments of DNA in this circular piece of DNA called a plasmid, which then we can introduce into our sort of lab-friendly workhorse strains, our typical E. coli lab strains. And basically within this E. coli strain, each colony would be expressing a small fragment of the total DNA that comes from one of the many microbes present in, let's say, a soil sample from the oil sands. So we can represent all this DNA in a host strain, but we can't represent it in its native strain where it's grown from, because we can't recover those isolates. And so in these kinds of libraries, it's basically a library of environmental DNA, and they can represent hundreds. In this case, this library represents almost 600 bacterial genomes. This is sort of an open science project. These kinds of libraries were created where soil was cloned from all different kinds of soils and made available to researchers as a library of DNA for us to go and mine out the interesting enzymes that bacteria produce, right? Scientists everywhere are hunting for enzymes that are useful. You think of the enzymes that are present in your detergents and laundry detergents that degrade proteins and fats and help your clothes get clean. Scientists are searching for enzymes for all kinds of purposes, degrading these pollutants, for example, degrading plastics, because we now know that microbes have evolved the ability to degrade plastics. So we're searching for those enzymes. These are an excellent tool to search and mine through this vast library for the individual genes and enzymes that have these key activities. But how do you screen through thousands of genes and thousands of individual isolates? You need a high throughput test. You need something that's fast and simple. And that's what this biosensors provide for us, where in an earlier study, in our first studies with this, we basically picked 5,000 colonies from these plates. So these colonies were then grown in individual wells in these microplates, and we pick stacks and stacks of these colonies so that you have, roughly 100 colonies and strains per plate. If you have 50 stat plates, you have basically 5,000 colonies. We grow each of those colonies and cultures in liquid with additional naphthenic acids. We can grow them as short as 1 day. And the next step is we add our biosensors. It's a co-culture. So we're adding our oil sands biosensor into this E. coli culture, and we're giving it a few hours to incubate. And then we take a luminescence reading. And what we see is most wells light up from the biosensor, giving us a strong, consistent signal that says, yes, there's still naphthenic acids here, and the biosensor produces lots of light. What we look for is the dark well, the individual colony that doesn't produce any light because that culture has degraded the naphthenic acids. And the biosensor is telling us there's no more naphthenic acids here. So here we can do this sort of needle in the haystack approach using this very simple assay. This was done in a few weeks. This would take a lifetime to do, literally, if you did this using conventional analytical chemistry methods. Here's just an example of the data from 1,400 strains that we said which of these strains are capable of degrading naphthenic acids. Most of the strains have this strong signal that's very flat. Here we're interested in the few strains where the signal is very low. These strains are degrading naphthenic acids, and the sensor is telling us. There's a few anomalies where the signal goes up in the biosensor. We're not exactly sure what happens, but for us, we're more interested in what is happening in these strains. And then we go and sequence the DNA that we recover from those strains so that we now know the exact sequence of that single fragment of DNA that came from some unknown microbe that was present in the soil. And now we have a bioinformatic challenge. We have about 40 genes that we're trying to figure out somewhere on here. These are the genes that enable this bacteria to degrade naphthenic acids. And now we're trying to figure out what they are. So we're getting to the end stages, and it's been very successful. So I have 2 more slides. First, I would like to thank my lab members. My current student is Tyson Bookout, and he's preceded by Steve. We just hired a postdoctoral fellow, and we've had numerous undergrads, including an undergrad from Athabasca University, Jenna. These are our funding sources and our collaborators. So we have an expert on the analytical chemistry of OSPW, an expert on bioremediation and an actual oil sands microbiologist, and our collaborator that helps us sequence all of our genomes. And with that, I can take any questions.
That's awesome. Thanks very much, Shawn. Everyone can clap in the background. There's a couple of questions in the chat already, and they're both, I believe, from Bruce Jackson. And Bruce, I'm not sure if you want to ask them out loud or have me read them, but I'm happy to read them out for you.
I can see them.
Can you see them? Okay, yeah, sure. Go ahead, Shawn, if you want to.
That's a good question. There are sulfate reducers, and sulfide gas is a problem. But in this case, I don't know if these sulfate reducers were the ones that made disulfide gas. If that were true, you'd be trading methane for sulfide gas, so that might not be a win. But I'm not sure. The sulfate reducing bacteria are also very complex, and that's not my area of expertise. But that's something I'd actually like to look into and get back to you with a more detailed answer. There's another question here.
Another question. Yeah, maybe I'll read it out for the audience, but would the new sensors identify what was happening in the Toms River in New Jersey, Toms River, New Jersey, Dow and Ciba-Geigy chemical? Sorry, I didn't pronounce that right. It seems the chromatography is still important.
I'm not familiar with that particular sort of environmental issue. Definitely the analytical methods are the norm and the standard. And they would, if you're tracking down something that's unknown, that would be the use. You would use conventional methods. Our system, you need to know what you're looking for. So we could make biosensors for numerous pollutants. They've been developed for decades. They've been developed for numerous compounds. They are hardly or very seldomly used in the industry. Arsenic, there is an arsenic biosensor that's starting to be used, where arsenic contamination of various groundwater and things like that. So the hope is that we're now after decades of developing these, that we can actually start to build a good argument to start to use them for environmental monitoring, but I'm not too sure about that particular case.
My question would be as well, do you think they'll have this fancy microbes cleaning up the tailings ponds before they release them in 2023?
Oh, they're not releasing anything by 2023, to my knowledge. They can't release anything until the water's been treated. The tailings ponds are just the storage vehicle now. There is some degradation that happens in a tailings pond, but it never gets to really low levels. Mm-hmm. So they are working on other strategies like an end pit lake or a constructed wetland. So an end pit lake is where you dig a lake and you put in, say, 20% of the volume is the tailings water and 80% of fresh water, and that you create a more hospitable environment for microbes to do the degradation. Or a wetland or a constructed wetland is filtering the water through a wetland that's populated with very specific plants, the microbes from the tailings ponds. And in that wetland environment, they are doing a natural bioremediation with the combination of plants and bacteria, so that when it filters through, the degradation has taken place. And that would take place, that would be good for all degrading all the compounds and all the organics in the tailings, not just naphthenic acids.
Oh, I see Carmen's got a question for you. Maybe I'll read this one out again. Thanks, Shawn. Are you able to trace the genetic fragments in your libraries back to bacterial species?
I'm going to say sometimes you could. What we're noticing is that this DNA doesn't necessarily look like too much of the other DNA that's sequenced in the databases, which might not be surprising because it's coming from a microbe that's probably never been cultured. Therefore, it's never been sequenced. So it's hard to trace it back to a microbe that's never been sequenced before. That's the short answer.
Yeah, I have a bit of a follow-up question to that, I guess, Shawn. And in that one figure that you showed to Carmen's question of piecing some of that sequence together, it looked like there were 5 or 6 genes that you were able to identify functionally. And so, yeah, really the other ones, I guess, is lack of data in the database. Is that correct? Are there any other methods using other databases that you could at least predict function?
So if you take the sequence that we generated and you annotate it, which means you try to predict what genes are there, and then you search for homologs, you often don't find reasonable homologs. So just the gene prediction is difficult. Yeah. So we probably, well, not probably, we will use another method to genetically map what genes in that 35 kilobase region. It's not that big. We can, by mutational studies with transposons, we can determine which ones there are necessary. So we can do a functional screen, again, using our biosensor. It's all hinged on using this really simple test that can tell you are the naphthenic acids degraded or not. And so we can functionally identify the genes within that small cluster to prove which specific genes are doing the job.
No, that's good. Thank you. So I'll ask you one other question, Shawn. What's the likelihood, or how soon you think it would be possible to use one of these biosensors, the naphthenic acid detecting biosensors, in the field? What would that look like? would it be possible for communities to use that for kind of direct monitoring?
It should be possible to do that in the near future. One strategy is there is a biosensor company, a startup company in Calgary named FredSense, and they develop these in-field testing devices where they use the biosensors in the field. So they build a device that should be adaptable for any number of biosensors. So one possibility is to collaborate with a company like that. So we give them the biosensor and they have the device and they combine the two technologies. Or there are other ways to make it field ready that are not that difficult. And so I would say within a few years, this could be something that if there was a significant interest, it could be used in the field.
That's good. Thank you. Are there any other questions? Oh, here's one more from Vive. Given the impact of this find, oh, we've got another, scroll up here. Given the impact of this find, the patent, congratulations. How could we develop a program around it?
I think I've already developed a research program around it. So maybe you might have to elaborate, Vive, what you mean by
Vive, do you want to
program.
turn on your mic and camera?
Thanks, Shawn. This is so fascinating to listen in. What I'm looking for is, yes, research program clusters are brilliant. Yes, you already are highly successful in that area. I'm curious about bringing these pieces of knowledge into academic programs. Of course, we do have courses, but is it possible for us to have a concentration, given the importance and the climate and the environment and all the concerns involved? I think it would be prudent on our part to conceive of a novel academic program that will benefit a lot of people, industry alike.
Yeah, that's a complicated question, but I think the kind of microbiology that this sort of microbiome research basically, and whether it's in environment or health, is already having a huge impact, or it's about to have a huge impact. So how do you make it real? That's a really, I mean, I wish I knew the answer to that. I'm looking forward to getting our research published in the near future, and getting it out to other researchers to use and to see how useful and beneficial it is and find other applications. So that will be a big start, just to see if other researchers find this stuff useful. But this whole area of synthetic biology, which is what I think this line of research falls into, where you're engineering microbes to provide a useful service, I think that's already a huge area of focus throughout the world, and it's generating a huge number. I actually have another slide here on this sort of topic, Vive, that you might appreciate. A huge number of industries where I like to say that the microbes have an opportunity for service. But so in all these different sectors, you have these biotechs being generated out of this kind of microbiology. So not just in the environment, but in sustainability, where everything currently is produced from petroleum, well, non-petroleum based building blocks that come from microbes could be used. All kinds of biomaterials, biodegradable plastics. Though there's a huge food industry where animal-free foods are being produced, like milk-free cheeses, all kinds of protein sources, etc. Lots of medicines are being engineered to be produced in microbes and yeast and bacteria, as opposed to purifying them from plants or their normal sources. So, and then the probiotics and the gut health is a huge area where there will be numerous applications of engineered microbes or microbes that we identify through this microbiome science, that these are going to have a huge impact in health and in biotech and in environment in numerous ways. I don't know if that answered your question.
Oh, this is unbelievable and brilliant synthetic biology. I've heard of this term, but I didn't realize that it percolates into pretty much everything we do here. Again, my question, it's not really a question, it's a comment. I think we should seriously consider developing a program, given the expertise we already have and the research team that you have built already. It's very natural for us to find a way to program it so that it can benefit people around the world, not just the labs, not just researchers.
Well, right now I do a lot of the work at the University of Calgary. So we are discussing around, utilizing the lab space in Athabasca and building a research culture there, a stronger research culture that could be doing these kinds of things there. It's not that any basic laboratory can do this kind of research.
Mm-hmm. Excellent.
I mean, if I
Thank you.
may add, Viv, I think, the work that goes on here has, it definitely has crossover in a lot of programming areas, which, that around bioinformatics, computational biology, even possibly some data science, I believe. So I think, within those types of concentrations, this research could be supported and emerge and develop more widely, but certainly, yeah, there's great opportunity, I think, to do more, especially in particularly with the environment and with the communities that we have connections with. And I'm thinking particularly in Northern Alberta and our relationship to Northern Alberta and what we can offer in that regard.
Yeah, I fully agree. Thanks, Shawn. This is wonderful.
Thanks, Viv.
Yeah, I think if there aren't any other questions, I want to thank you very much, Shawn. It's just a really interesting talk. And I think just a great topic to cover for science outreach. I think it gave a lot of interesting detail about the microbial world, which some of us are fascinated by that are microbiologists, but it's nice to see that shared with everybody else. And so I thank you very much. And I thank all of our participants for attending. It was another great event. And thanks so much to Linda for her organization and all the members of Science Outreach.
Thanks everyone for attending, and thank you, Shawn, for taking time out of your busy schedule for doing this. Just an after note, we do have a Christmas bird count coming up on December 27th. In January 25th, there'll be a talk on dinosaurs, and February 22nd, a talk on methane. Bioconversion of methane to biodegradable plastic.
That's cool. Thanks, Linda.
Thanks, everyone.
Thank you.
Thanks, everyone. Have a good night.
Take care.
Take care. Thank you. Bye-bye. Thanks, Shawn, again.
Okay, so I'm signing out now.